Okay, folks, so here we go. We're going to be annotating hemophilia proteins and genes. Um, First thing you want to do is look at the images. So we have here, it looks like it's a scrape, right? So maybe someone scraped their knee. And here, what do we have here? Well, these are actually every single one of these little red circles are proteins. And they're bonding together to form a clot or a scab. And that's what that is. And then we also have a protein here. So there are 13. This is a protein. So we've been talking about protein, and this is not a model. It's a model, but it's a great representation of what a protein looks like. And as you can see here, there are 13 different types. And this represents, this represents one of 13. So we have hemophilia proteins and genes. You fall off your bike and you scrape your knee. Blood drips out, but as soon as the scab starts to form and the bleeding stops, sounds simple, doesn't it? However, if you could zoom into to see what's happening on a molecular scale, it's not simple at all. Your blood includes 13 different protein molecules called clotting factor. And that's what they're referring to over here. When you injure yourself, these clotting factor proteins all connect and work together to form a solid clot known as a scab. If you were missing even one of these 13 proteins, the clot would not form and the dangerous bleeding would continue. This is exactly what happens in a disease called hemophilia. People with hemophilia are missing one of the proteins needed for blood clotting. And so for me, this here is important. So hemophilia is a disease. Right? Dangerous bleeding continues. So it basically means that you have blood and that in which you stop bleeding. You just stop bleeding when you have these 13 proteins and you get a scratch. But if you're missing one of these 13 proteins, you're going to have hemophilia, and it's a dangerous disease. So people with hemophilia are missing clotting proteins because of a protein with their genes. So you are born with hemophilia. You're born. It's not the food you eat. It's not because you don't exercise. You are just born with hemophilia. And it has to do with your genes. Okay. And the genes, as we know, carry instructions to make different proteins. There may be several possible versions. And how many people have this? Think to yourself, well, let's check Google really quick. How many people have hemophilia? Or what percentage of the population? One out of every 5,000 male births, based on a recent study, about 20,000, as many as 33,000 males in the United States are living with the disorder. So 33,000 people, 33,000 males have this disease. And here's a picture. So a father does not have hemophilia XY. Mother is a carrier of hemophilia. 50% chance that a son will have it. 50% chance daughters will be a carrier of the hemophilia gene. So is this telling us that only men can have hemophilia? That's interesting, right? And the results tell us here how much of the population has hemophilia. 1 in every 5,000 are born with it, and 400,000 people worldwide are living with hemophilia, and 20,000 are living with it in the United States. So about 20,000 people. And it says there what gender is most affected. Looks like male is greater than female. Interesting. So 20,000 people in the USA have this disease. All right, let's keep moving on. More pictures. Awesome. More pictures. So we have genes and proteins. We discussed this before. So this is a cell. And this is the nucleus. This little dot that you see there. And we have chromosomes. And I wrote here, chromosomes. You can have chromosomes from your mother, chromosomes from your father, and they're intertwined. And you have the protein that lives inside of the DNA. And the gene is what gives the instructions for your trait. Like, your hair color or how tall you are. All right. So an organism has 2 copies of each gene. 2 copies of each gene. So here's copy 1. Here's DNA, right? So there's 1 copy. 2 copies. These gene copies can either be the same version, homozygous, or a different version, heterozygous. We haven't seen those words, so let's highlight. Homozygous, same. Heterozygous, different. If the gene copies are homozygous, just one type of protein is produced, since both gene copies give the same instructions. If the gene copies are heterozygous, 2 types of protein are produced, since each gene copy gives a somewhat different instruction. So that sounds complicated. I agree. If you were to listen to this here, but you might have gene copies that are the same, or you might have gene copies that are different. So to explain what's happening here, the best thing to do is to look at this right here. So this is a gene copy. This is also a gene copy. This could be the gene copy of a person. This could be the gene copy of a different person. And you carry the same gene copy here. So it would be homozygous when it's the same. When it is different, then it is heterozygous. An organism with two gene copies that are the same are homozygous. These are gene copies, right? While an organism with two gene copies that are different is heterozygous. You might get confused with this word organism. So an organism, this could be an organism of a giraffe. A giraffe lives and it breathes. It could be a lion. It could be a butterfly. It could be an insect. It could be a human. It could be a reptile, like a snake or a crocodile. These are all organisms. Okay? It lives, it breathes, it dies. They're all organisms, two gene copies. So where were we? Over here. We're talking about people with hemophilia have a version of the gene that doesn't have the right instructions for making clotting factor proteins. Remember, when you see the word clotting, think about scab, creating a scab. If your body doesn't create a scab, like look at my arm right here, I got this cut, and this is a scab that has formed. Okay, a clot. Clotting is formed, blocking the blood from coming out of my arm. So this is a scab. Without the right instructions, their cells can't make that protein, or they make a protein that doesn't work. Without functional clotting factor protein, the proteins in their blood can't make the connections needed to form clots and build scabs. So all 13 proteins are needed. All 13 proteins are needed to make clots or scabs. So I have 13 of these in here. In here I have 13 of these protein molecules working along with platelets. We'll talk about those later. That help create a scab and prevent you from bleeding. Let's see here. Where were we? Healthy people have at least one copy of the gene version that provides the right instructions for making all the clotting factor proteins. Being able to form scabs may seem so basic that it's strange to think that it is a genetic trait like having brown eyes or having freckles. So, brown eyes, freckles, they are what? Traits. Traits. Oh, and going back, this is the main idea for that paragraph. And usually it's the first sentence. Usually it's the first sentence or it's the last sentence. And right here, let's see here. The ability to form scabs is a trait that is determined by proteins. To have that trait, you need to have those proteins. And to make those proteins, you need to have the genes that give your cells the instructions for making them. So, yeah, let's say it's this. Treating hemophilia with proteins. So, even though hemophilia is a serious disease, most hemophilia patients can live relatively normal lives if they get proper treatment. So it's all about treatment. Okay, maybe medicine. Hemophilia is caused by a missing protein, so doctors treat it by replacing that protein. Doctors inject clotting factor protein into a patient with hemophilia so the patient's blood clots the way it's supposed to. So that's what they do. They inject people with clotting factor proteins in order for the body to be able to create clots. Injecting the protein into the body does not change the protein the body produces. Instead, it supplies the protein the blood needs to clot properly. People with hemophilia need to have the missing protein injected into their bodies throughout their lifetimes because the injected protein will be used up. The patients then need to be injected again for their blood to continue to form blood clots properly. So what is the main sentence of this paragraph here? That was a very long paragraph. Actually, we already highlighted it. That was a long paragraph. So what's happening here? We have scientists are making progress in treating hemophilia and other genetic diseases. Okay. For many years, the only way to get clotting factor protein was to separate the protein from the donated blood. However, it takes a lot of blood to get a little bit of clotting factor protein. So today, scientists can make lots of clotting factor protein by inserting human genes into hamster chromosomes. Interesting. So scientists insert human genes into hamster chromosomes. If you put the human gene for making clotting factor protein into the chromosomes in the nucleus of a hamster cell, the cell makes the protein, even though it's a hamster cell. The cell makes clotting factor protein that's identical to clotting factor protein made in human cells. As long as the cell has the gene with instructions for making clotting factor protein, the cell will make the protein. Interesting. And let's highlight this right here. Today's scientists can make clotting factor protein by inserting human genes into hamster chromosomes. That is very interesting. Gene therapy. Hemophilia can be treated by using clotting factor protein, but it can't be cured, for now at least. Scientists are researching possible cures for hemophilia, cures that would get the patient's own cells to produce clotting factor protein. For this kind of a cure, scientists would have to figure out how to fix a patient's faulty gene for making clotting factor protein. If the faulty gene were fixed, the patient's cells could start making clotting factor protein naturally. Working to fix or replace genes is called gene therapy. Gene therapy. I think that's what's important here. Working to fix or replace genes is called gene therapy. And it says here it can't be cured yet. So in gene therapy, we are fixing or replacing genes. And that's all, guys. Good work today.